Hi, 7th graders from Ames Middle School. We got lucky today, and instead of just getting to chat with Ruth, who's standing over there casually watching, we're going to get to chat with some of the people in the lab who actually help make the GMOs here in Ames.
I just said these guys do GMO work here in Ames. Well, that's pretty close to accurate because they do select for specific traits and try to get them in soybeans. They do it through breeding, so it would be more accurate to call them breeders than genetic modifiers.
Could you introduce yourselves?
My name is Danny Singh. I'm glad you're watching this video. We love science. I hope some of you will take science as a career. I'm a plant scientist working at Iowa State University.
Sweet.
Hi guys, my name is Jonathan Shook. I'm a student with Dr. Singh here, and I'm working on trying to identify exactly what's causing the differences in how soybeans perform. So looking at the molecular causes of that and finding out what different genes are doing within the crop.
Sweet. So you figure out what genes do what.
Exactly.
Awesome.
Hi, my name is Kevin Chiteri and I'm a student with Singh. And in addition to what John said is that so we just help empower the plants so that they can survive when it's too hot, when it's too cold, when they are affected by insects.
Yeah.
Yeah.
So you help the traits be better for their environment.
We're
Help
trying
the
to make
traits survive.
soybean be more productive.
Yeah, so the farmers can make more
So,
money
yes.
and we can get more food.
They're environmentally conscious.
Sweet. So we're in a lab right now. There's a whole bunch of stuff going on. And the lovely people we were just introduced to are going to kind of show us around a little bit. So let's take a look.
So generally speaking, the lab is a very active place. A lot of people are in the lab working on different projects.
Group work is a thing, guys.
Group work is a thing. We work as a team. There are lots of different people in the lab. So as an example, this would be what would be a workstation. So a person, a scientist would be here. They would be doing their research, conducting their research, working with different aspects of the plant cells. So they may be looking at DNA, they may be looking at RNA, they may be looking at protein. So a lot of equipment and instruments, and I'll have John and Kevin explain a little bit more about some of the things that are going on in the lab.
Sweet.
Turns out we don't have to go all the way down there. So this is what's called a gel electrophoresis setup.
Whoa, that's a big word.
Right. So this is the PCR. So you're putting DNA into a gel, and then putting electrical current through it so that it moves throughout there. Small pieces are going to move more because they're more strongly pulled by that electric current, while the big pieces stay back further.
So it's kind of like a race.
Exactly.
And they're running a race, and the little ones have less to move, so they win that race.
Yep.
And the electricity, so we have some cords hooked up to this. The electricity is helping them run that race and push the little ones, right?
Yep. So what you're going to get in the end is you're going to have distinct bands that you can see where you can visualize that DNA, and you get bands running across for each lane that you're running. So you can run maybe 30 at a time and be able to see which combinations of alleles that are within each one of those lines. So you can identify if something's a hybrid or if it's purebred or inbred.
Yeah, okay. So those are more things we learned about in class. So what else is there?
Alright, when John also said that, so you visualize using the machine, right? But this right here
So this thing, what's it do?
is the machine that you're able to visualize the gel.
So
Once
this
it's
helps
the
us
gel-
see stuff.
Yeah, you can see
Okay.
because you can't see it just with your bare eyes.
Okay.
So you get in this machine and then you can be able to visualize the bands.
So do we see that twisted ladder shape?
No, you won't see this, right? Your teacher just told me you won't see this. You just see like bands.
Okay, why can't we see it?
They're too tiny for
Too
our
tiny
eyes.
for our eyes.
Right.
Yeah, super duper small.
It's pretty small.
Yeah. Okay, sweet.
Yeah. So what else we wanted to show you, because this is a molecular lab and this is the molecule. So it's called the molecular lab as basic things that you must have. And one of them is the fridge, which is at plus 4 degrees Celsius.
So that's chilly.
So that's 40 Fahrenheit.
Yeah,
So
40
kind of
Fahrenheit.
like your fridge at home.
Right.
Kind of like your fridge at home.
Right. And then
So what's this one?
and then we have a freezer here. You thought Iowa is cold, but this is like, can you see that? Negative 79 degrees Celsius. That
A
would be
little
like-
bit.
Negative 112 Fahrenheit.
Exactly.
Wow, negative
Right.
112.
Right.
And we think today is cold.
Right.
Yeah.
So we do store here our samples because they are sensitive to temperature changes, and so once here we know they're well stored.
I wonder what kind of ice cream you could make in a negative 80 degree freezer.
I don't know. Maybe
Yeah.
you guys make and bring us right here you see is the cleaning station.
Mhm.
So just like at home, we have a place where I'm sure you guys know what this is, right?
Maybe.
Yeah.
We don't get to do very many labs.
Okay, but that's
Seventh
okay. So
grade.
you have a station where you clean your stuff all the time.
Mhm.
Yeah, and many more instruments that we have in the lab, and we'll be happy to host you guys to come and show you more.
Sweet. So yeah, thank you guys so much. Let's say goodbye to our 7th graders.
All right.
Bye.
Bye.
Hi.
So, this is Ruth McDonald.
Hi.
And I've been told you know a lot about GMOs.
I know a little bit. Yep.
Oh, okay, cool, sweet. Because we have some questions for you today. First of all, can we ask a little bit of a question beforehand of what exactly your role here is at Iowa State? What exactly your job is? What
Sure.
exactly you do?
Sure. I'm a professor here at Iowa State University, and I am in a department called Food Science and Human Nutrition. And so our work in that department is to understand how food helps your body. And we also understand what food is, and we make different kinds of food. You might see behind me on the shelf there is some popcorn. We have one of our students that graduated, and she works at ConAgra, and she made that popcorn. She created that variety of the microwave popcorn called Boom Chicka Pop.
Sweet.
And so that's what some food scientist people do as well.
Okay, so you would know pretty much anything about if the food is safe, how it might affect you, and where it came from, right?
Correct.
Sweet. Okay, cool. I know there's a lot of questions in here, so let's pick
Okay,
one.
Let's pick this one.
Let's pick that one. Okay. How much money does it cost to genetically modify something?
That's a good question. It costs a lot of money to genetically modify something, and that's because it takes scientists
Mm-hmm.
to first understand what the gene art is that they want to put into the plant or the animal, and they have to carefully study that to make sure it does what they think it does. Then they have to actually take the time to create the seeds for crops or
How
for
do they create a seed?
well, they have to take a seed that exists, and then they have to insert the gene into it. And they use tools to do that. Sometimes they use a tool that will actually just like inject
Just
it.
literally like a needle. You
Mhm. And
put the
sometimes-
DNA in the seed.
You do, yeah.
Huh.
You just do that. And
Neat.
sometimes you would put it in another way, like it would absorb into the seed. There's
Okay.
other ways to do it.
Okay.
But you have to get it in there, and then you have to show that it's there. You have to
Okay.
show where it is. So you have to do all kinds of studies in the lab to kind of take the DNA and separate it and look at all the different genes as they're expressed. That's the easy part in the lab, actually, because the next thing is you got to take it out into the field, and you have to see how the plant works in the field.
So you have to grow the
You
plants.
have to grow the plant because sometimes they don't grow very well, right? And so no farmer is going to want a corn plant that doesn't grow very well.
Mhm.
So you have to show that it still has the ability to grow and that it does produce what you want it to produce and that it actually has that trait in it. So it takes probably 10 years to
10
go.
years
Yeah,
for
from
all
the
of that
lab
testing. Think
all
about
the
all
way
that
through.
money.
It takes a
All
long
of that
time.
money.
A lot
Lots
of
and
people,
money.
lots of science, a lot of
Yeah, 10
oversight.
years of paying hundreds of people
Yes,
full-time salaries.
right.
Man, do the math.
Right.
That's a lot.
Lot of money.
lot.
Yeah,
Would you
yeah.
say it's in the millions?
I would say it's in the millions.
In the millions.
Hmm.
Wow. Okay, next question. Okay, what types of food can kill some insects and how? So some foods can kill some insects. We learned that in the video. So how and what kinds of
Yeah.
food do that?
So the one thing that I'll talk about, just as an example, is BT.
Okay.
So that's a Bacillus thuringiensis is
That's
what
a big
BT
word.
stands for. So bacillus is a bacteria,
okay.
right? And
so
that's
it's
something that
bacteria.
you know what bacteria is, right?
Yeah.
The thuringiensis part is just the name of it.
Bacteria.
So bacteria lives everywhere,
Yeah.
right? And this particular bacteria is in the soil.
Okay.
And scientists figured out, actually farmers did way back in like the 1800s, they figured out that this particular bacteria produced something that killed this particular kind of worm that was eating
Okay.
corn roots.
Okay.
The corn root worm borer. Borer is the name of the bug. And so they figured out that if they got this particular bacteria and they grew it up and they could get this protein out, that they could prevent this pest from damaging their crops. So the BT toxin, which is produced by this bacteria, is what they put into the corn plant so that when the pests, the larvae start to eat the root of the
Mhm.
corn, they get that toxin and they die.
Okay. So bacteria lived in the dirt.
Naturally occurring.
Bacteria living
Naturally
in the
made
dirt.
the made
The
this-
bacteria didn't want to be eaten by the worms. So the bacteria made a chemical that would kill the worms if it ate the bacteria.
Right.
So you put the worm-killing chemical from the bacteria into the corn.
Right.
So that way, when the corn is eaten by the worms also die.
Gets the bacteria, gets the toxin, and then they die. And that toxin doesn't do anything to people.
Nope.
It's a different kind of
Yeah.
bug thing.
Just like, we can eat chocolate, but dogs
Dogs can't.
get sick.
Right. And then just also that toxin that they use in the BT
Mm-hmm.
corn also can be used as a natural pest resistance in organic farming. So it's very safe. It's used
Sweet.
in organic farming too.
Cool. Very good question.
All right. What's this?
What's that one? So this one asks. So this is about the video, and the video had some questions about how we can do lots of GMO products to help save the environment. So at the end of the video, the question specifically says, "Why don't we do the thing in the video, where we're trying to counteract global warming and make GMO crops that do pretty big changes, like a super tomato that gives you antioxidants. Why don't we do all of these big GMO changes?"
So I think the main thing comes to the first question that I just showed you, we talked about, right? The money, right? How much does it cost?
Mm-hmm.
So a company or some scientist has to have a reason to do what they're going to do, and they have to have some way to pay for it.
Mm-hmm.
So if you don't have a company that's willing to invest in that particular kind of product or that crop, then it can't be done.
Mm-hmm.
So we either have to invest our tax money from the government in doing some of this science, or we have to rely on companies that think that they can make money on selling the product once it's produced. So that's been the limitation, really, is
Money.
where we can pay for doing the science to develop these different types of crops.
Good question. Good question.
Let's use
Choose
this
the one with the art.
one. Yeah.
All right, this one has a few. Let's choose. Oh, this asks the same question again about why aren't we making superfoods? So
Yeah.
that's why. And then also, what does non-GMO mean
Good question.
in a
Good
bottle
question.
on the store? Yeah.
That's a
So
good
what does
question.
non-GMO mean?
So you've probably seen signs when you've been grocery shopping, right, that says non-GMO right on the label.
Mm-hmm.
It is, what that means is that it has no ingredients that have been genetically modified using the tools that we just talked about, using the
The stuff
molecular
in the lab.
biology stuff
Yeah.
in the lab, right?
Mhm.
It's very misleading, however, because you might see a non-GMO label on something like carrots or on peanut butter.
Mm-hmm.
There is no genetically modified carrot or genetically modified peanut
In existence.
in existence in
So
the U.S.
every
anyway.
peanut butter
So every peanut butter
is
is
non-GMO.
non-GMO.
They're just trying to make money.
They're really trying to let people know that this is not
Mhm.
in that category. But again, it's misleading because it sort of implies that GMOs would be bad,
Mhm.
right? You would want something that's not GMO,
Mhm.
and there's no reason to think that. So
It's like it's
very
still got
misleading.
DNA in it.
right.
DNA is DNA is DNA.
So be a wise consumer and don't fall into the trap of
Yeah. So if you see a jar of peanut butter that costs $3 and says non-GMO, and a jar of peanut butter that costs $2.50 that doesn't say non-GMO, they're the same thing.
pretty much.
So you're gonna buy the cheaper one because non-GMO for peanut butter doesn't mean anything.
Right. All right. So what's this question?
So in the video there was a picture of the watermelon that had all the white stuff because all of the rind because it had been modified to become the watermelon we know today through just breeding. And this question says, I didn't really understand the watermelon thousands of years ago. So how did the watermelon change
Yeah.
from a long time
That's a
ago?
good question. You could ask that about any food, right? So how did we get a watermelon that has all that red inside that we like to eat and little less of the rind that we don't like to eat? We got it through breeding because scientists would take a plant that has more of the red and they cross it with a plant that has more of the red, and then they would get a new plant that had
So it
more of
gets
the
redder
red,
and
and they
redder
kept
and
doing
redder.
that and doing that and doing that until you end up with the watermelon that we have today.
Thank you.